Hi, everybody. Welcome back to the I Am IMA podcast, a podcast from the Innovation Methodology and Application theme here at Newcastle University. My name is Andrew Fuller. I'm your host, and I'm very pleased to be joined today by Professor Joris Veltman, who's the Dean of the Biosciences Institute. Thank you very much for joining me today, Joris.
Thanks, Andy. It's a pleasure to join.
Excellent. I do appreciate you finding time for me in what I can only imagine is an increasingly busy schedule. I guess I wanted to start today just by kind of going a little bit back in time, going back in a time machine, if you will. And I'd like to take you all the way back. We won't say how far. Take you back to where you started in academia. So I've done a little bit of background research, and I'll be honest, I've been stalking your LinkedIn page somewhat. But as far as I can tell, it started with a master's and an MSc in molecular sciences back in the Netherlands. Is that right? Have I got that one right?
Yeah. So I studied at a very small university, an agricultural university in Wageningen.
I'm so glad you pronounced that, because there's a few words in here that I'm struggling with. But okay, a small agricultural university. That's an interesting.
Yeah, that's an agricultural university. It's actually very famous internationally, because lots of international students come there and learn about, let's say, modern ways of agriculture. And so I studied molecular sciences there, which was a combination of biology and physics and mathematics and chemistry, which was a great, it was quite challenging, but it was a great start, I think. And the good thing is that half of the time there, I had to do practical work. So it was really a lot of, practical work, which really allowed you to understand things like, DNA and that was, I think, very important if you want to do a lab-based career.
Absolutely, yeah. I couldn't agree more. I think a lot of the if you like it, if you can find out if you're good at it, if you like it at an early stage, it allows you to focus on and steer towards it. That certainly was the case for me. It's interesting because I also did some work. My father was professor of plant physiology down in Plymouth. I've done a lot of agriculture work. I used to work in a small university outreach from agriculture. So yeah, I like it. I always got on well with plants, but I don't do much with them except grow them now. But and from there you did a PhD. You moved on to, I'm going to have a stab at saying the department name, but it's the longest word I've looked at. It's otorhinolaryngology. Yeah, did I get that right?
Yeah, you did. Yeah, it's
okay. Excellent.
ENT department.
ENT. Yeah, I just like saying long words sometimes. Yeah. So it's a bit of a change from a small agricultural university to an ENT department. What prompted that shift?
Yeah, no, it was actually a joint PhD with the Department of Molecular Cell Biology and Genetics in Maastricht University. So this is where I did my PhD on the genetics of head and neck cancer. And that was a shift I felt after doing the master's, working with the pea plants and with the different fish. I felt that for my career, I wanted to go into medical applications. I did feel that was useful, that I wanted to have something of medical impact. And so I found a PhD in that area. And of course, whether you work with plants or whether you work with animals or with humans, if it comes to DNA, the technologies are the same. But there is a bit more funding for the medical side. So that's true. Was a good thing.
That's certainly true. So what was the title of your PhD thesis in the end? I couldn't find that for lack of searching. So what did it come out at? What did it come out as in the end?
You're asking me a lot of questions. I just turned 50, so I have to now go back and think about all these things. So this was about chromosome aberrations in head and neck cancer.
Okay.
So this was using the fluorescence in situ hybridization technology, which was very hip at that moment, and we did quite some work with microsatellite markers. So that was at that moment the biggest thing. But after that, I don't know if you want me to so that
please carry on.
Yeah. So after that PhD, I that was the moment when people started to do things like microarrays. They came up the end of last millennium, basically, and so I wanted to learn to work with microarrays. I like very much genetics, and there was a group in San Francisco that had basically developed the first microarrays to look at deletions and duplications in the human genome. So that's I managed to go for a postdoc there, and that was a great start of let's say my real career, I would say, where I use different genomics technologies to study basically, let's say, how where there are mutations or deletions in the genome that cause disease, and understand genomic mechanisms of different diseases. So, yeah, that's
Sure.
where I really started. I think the postdoc in San Francisco.
And those, I mean, I've heard rumors of postdocing over in the States being a very intense experience. Was that your experience of it as well? Was it a really intense kind of, like, the work day is very long and the work ethic is really intense? Was that the same experience for you, or
absolutely? I mean, it was. I think I felt during my PhD that I was not really put under pressure, and I think I needed that a little bit. So in San Francisco, I definitely had I worked at a competitive group, and they had this really new technology that I could work with. But yeah, it was very normal to work in the weekends, and but if you're there on your own as a postdoc without further commitments, it's also it's kind of fun to do for a period.
Sure.
I mean, I really enjoyed that time, and I learned a lot, and I got some good publications out. But then I did feel in the end that while I liked, it was very technological driven, the research.
Right.
The problem there was that when it came to, let's say, the application, in the end, there was a fridge where they had, let's say, 100 samples of, in this case, it was a renal cancer samples, and I used these, but there was very little clinical link. This was from a group in Germany. They got some samples, and I used the latest technology, but actually on a cohort of samples which was very poorly characterized. So I wanted, in the end, and I found that out when I went back to the Netherlands, to really apply real technology, but really to answer important clinical questions. And so for that, you really have to have a good clinical link. And I think that's often much better in Europe. So in America, they're often very good at technology, but then they often apply it on samples which are poorly characterized. And that's, I think that's something we can do better here.
Okay, yeah, it's certainly something we do. It's fair to say something we do very, very well at Newcastle as well. There's a really good link between the kind of the basic research and the clinical research and the translational research between the two. It's really good here. That's certainly my experience of working here for the last 10 years or so. So after that, after you came sort of cut your teeth on the technological side of things in America, there was a fairly long stint back in the Netherlands where you were associate professor of genetics. Is that right? So was that into teaching roles as well? Did you take on some teaching roles there, or was it still stuck? Was it still a lot of lab work and things? Yeah. So basically, I came back. I mean, well, I was only in the States for a year. Yeah. And I came back first of all to set up basically the microarray technology in Nijmegen, and I was head of a microarray facility as well as a postdoc setting up my research. And then I basically developed my research career there. I got my first grants, and then became professor there. After it's a 16-year period, so a long period where I used genomics technologies, moved from microarrays to next generation sequencing, and applied this mostly to rare diseases, to unravel the causes of all kind of syndromes for which people knew that they were genetics, but they could not really, do anything until there was this kind of unbiased approach where you could just look at a patient's genome and compare it, for example, to the genome of the parents. And yeah, that was really my breakthrough, where we found these de novo mutations. I don't know how much you know about that, but that's my biggest, let's say, contribution to science, where we, one of the things that if you have very severe early onset diseases, they are actually selected against. So you basically, you have a child with a developmental delay and a heart problem. That child will very likely not reproduce him or herself. So the question is, how come that these diseases are still so frequent in the population? And that's basically what we've been studying by comparing the genomes and the exomes of these children to the exomes of their parents, because these parents were healthy. They got a child with a severe disease. And what we found out is that there is these kind of de novo germline mutations that come in each one of us. And we each have 50 to 100 of these mutations. And these mutations have not been selected in the, let's say, the
or over evolution. So they actually can often be quite severe and damaging. And they are a major cause of developmental problems. That's basically what we could see using these unbiased genomics approaches. So that's really something where technology has advanced our field and has really now is used routinely in diagnostics. And that's quite nice to see.
And how do you think, like, I'm always interested in the barrier of entry is something that's on my mind a lot lately. How do you think the barrier of entry for kind of PhD students or postdocs wanting to do that kind of next gen sequencing type of work on their samples, it's got to be coming down, right? The ease of access to that sort of equipment and the ease of access to sample cohorts for where you are and the questions you can answer with it and ask with it, it's got to be becoming easier and easier. I imagine that was a lot harder to do only a few years ago, 5 years ago, 10 years ago. It was a lot harder to get those sorts of systems in place, and by the sounds of things, you've had to pretty much set up a facility yourself to actually get the work done. So do you feel like the barrier of entry is coming down significantly since you did that? Or?
Oh, absolutely. I mean, these technologies, I mean, that was really, we were really pioneering that. There were a few groups in the world who did this. And then, this was really where genomics technologies was still special. And I think, I often give talks where I talk about how genomics has become an industry.
Sure.
And of course, nowadays, yes, there's all these kind of companies that provide next generation sequencing services for you, and we have a fantastic genomics core facility here in Newcastle, led by John Coxhead, and nowadays it's quite interesting. As a PhD student, you can basically hand in your DNA sample, and then a few weeks later you get your exome back, and it costs you a few £100. So that is something that, of course, that's an advance in the field that we cannot imagine. I have to say, I'm very glad that I've gone through all of these things. So basically working with a microarray with a few hundred probes to a microarray with 1 million probes to next generation sequencing with let's say, looking at a few genes to looking at the entire genome. Because one of the challenges, I think, for many PhD students is to really go, they basically come in, they immediately get this overload of data. And I think that's learning to work with that and to prioritize variants and to deal with it and understanding it. That's a little bit of getting used to. But on the other hand, of course, it gives you an enormous amount of information that, if you use it in the right way, can be fantastic. And of course, nowadays we say, well, you need to combine that then with other techniques, and of course it's less about the DNA technology; it's more about what do you do with it. How do you interpret the data, and that's still there's still a lot to learn there.
I think that certainly on the large data set aspect side of things, like the interrogation that you can give, the effort it takes now to create a very robust, very large, like well-characterized data set is lower than the effort it takes to fully analyze that data set. I think certainly from my side of things, from the flow cytometry side of things and mass cytometry and all the tools we have at our disposal to create bigger and more powerful data sets with more probes and add more and more. And the amount of work you have to put into then analyze those things fully is phenomenal. And you have to then have specialists who know how to analyze that and be able to pull out the plots and the figures and answer the questions that you have. It's almost the technology to create those data sets is now so readily available, but the skill set is it's still up and coming for the amount of skills we need from the bioinformaticians and the informaticians that we've got on hand here. But there's certainly an expansion there that needs to happen as well to keep up with it. And it's certainly to my view anyway, because it requires us all to be big data scientists as well as big data producers, and that's
I mean, I think what I see is more and more important is that you need global collaboration, right? So what we see in the disease. So I work now on male infertility, and so far, if you look at literature for that, the genetics of that, you see individual groups reporting on 100 exomes or sometimes on 1,000. And then finding a few genes where there is some mutations, and some of them are recurrently mutated. So you can say, okay, probably this gene does something with this disease. But what you see happening in most disease fields is that you get these big consortia where people say, we need to get the data together from everybody working in this field, because that's the only way in which, because we are looking at diseases which are genetically heterogeneous. So you need to combine data sets, and 1,000 is not enough. 10,000 is not enough. So it's, I think what in my field of genomics, you really need to try to set up very big collaborative initiatives, and because that helps you then to find all these rare mutations, which together actually explain a significant fraction of a disease. Sure. And so that's, and actually I'm quite happy with that, because that's still something that I'm pretty good with. I'm not the best bioinformatician. I can't program a single line, but I can bring people together, and that's still the fun part. It's trying to get colleagues to share their data. And sometimes clinicians who are used to the old-fashioned way of just getting their own publication on a few hundred samples, but it's, convincing them that it's really important to work together now. And yeah, that's where I see a lot of the benefit going.
I think correct me if I'm wrong, but you've still got a research group over in the Netherlands as well. Is that right? Have you got both now, or did you have both for a time? I couldn't quite figure that one out.
Yeah, no. So I finished that. So I had the first 3 years. I still had funding from the Dutch scientific organization, and I had a group, a small group in Nijmegen in the Netherlands. But that finished basically July this year officially, because the last grant ran out in July. So it's good because, as you can imagine, combining the role of dean with a research group is more than enough. And
that was one of my questions.
I completely focused on Newcastle.
Yeah. I was one of my questions was, I was wondering how you did that, how you balanced those two roles, because I know you went from director of the IGM over at Centre for Life, because I think you and I overlapped by about 6 months. I think I left working at Centre for Life just as you started, or certainly collaborating there. So I've got to ask, like, how did you manage to maintain that research side of things over in the Netherlands and do the work over here? But was that a challenge? Obviously it was a challenge, but how much of a challenge?
I think it was. When you after 16 years I decided to move to Newcastle, of course that it was a challenge for me, but also for my group in Nijmegen. And so I had a number of PhD students, some of whom had just started, and that was of course a challenge. So I didn't bring people with me to Newcastle. I started completely from scratch, but I decided to keep joint appointments. So every month I basically went back to Nijmegen. This was pre-pandemic, so it was easy to travel. You fly in an hour to Amsterdam and you take a train to Nijmegen, which is another 1.5 hours. And I basically spend 2 days every month in Nijmegen to work with the PhD students who are. I'm very impressed with these students who somehow allowed that. And of course, I had a lot of also online discussion and supervision with them. But yeah, they managed to with also additional supervision in Nijmegen, of course, it's still there. They managed to finish their PhD. So I had already 2 successful PhD vivas from Nijmegen this year. There's another one coming. So they are basically now all finishing, and I'm very proud of them. But yeah, that was only possible because of let's say, one, that I was still flexible to travel, and the other that the students were quite independent. But yeah, I definitely don't want to do that too often. So.
No designs to establish a new group somewhere else across the other side of the world.
No, not at the moment. We're very happy. We have a young family, and we're very happy here in Newcastle. So we, we just had also the first 5 are here from a colleague here, so Kumara, who defended his thesis on Monday, 5 with minor corrections. So we're very proud of that.
Congratulations, that's excellent. And
I know there is now a lot of new colleagues. So I will definitely focus on the Newcastle role, and that gives me great pleasure. I'm very happy to, the research is still my big passion. So I have to say that I definitely want to keep my research group. And like anybody else, I'm looking for funding and all these things. So that still keeps me busy apart from my role as dean.
And of course, as a father as well to a young child, how is your young child getting on? I saw a very good photo. I saw a good video of him training with you for the Great North Run on Twitter.
Yes. No, he was a little bit ill the last few weeks, unfortunately.
I'm sorry to hear that.
But he's recovering. But on Saturday he slept well, so I could still do the Great North Run on Sunday.
Good.
And it was really good. It was really
excellent.
Good. Yeah.
Excellent. That's really good to hear. I'm sorry he was unwell, but I'm glad he's doing a little bit better. So I just wanted to shift a little bit, because I wanted to get your. I know you've been dean of NUBI, the Biosciences Institute, for a little while now, but right at the start of your appointment of that, we kind of got hit with a world-changing event, right, with the pandemic. I know I've mentioned it a lot on the different episodes of podcasts. I haven't done a podcast about it, but I have kind of brought it up to people because it is of interest to me and others, I think, how different levels at the university had to deal with that. So couple of parts to this question. When you took on the role of dean for NUBI, what was your role what was your vision for NUBI, for Biosciences Institute, when you started that? As like an overarching thing, it doesn't have to be too specific, but where did you see NUBI going, and how did that then change when we had to deal with the pandemic? How did that pandemic impact your overarching vision for NUBI?
Yeah. I mean, what I very much liked about, and still like about, the new structure is that basically this idea of we're not individual silos as institutes, like we were, to be honest, a little bit before, but we really try to work together much more jointly with a very big focus on team science. And I think that's with the institutes, but then also very much with the research teams working together. And of course, within NUBI, we have fantastic basic science, world leading, but we also want them to think about, what kind of applications can we have more in the translational area? How can we get our technologists, people like you, to work with our colleagues to really advance fields? And I think that's basically where I see the area of where I can help out. So I'm not an expert microbiologist. I'm not an expert neuroscientist, and it's very challenging to lead an institute with so big diversity of topics. So my vision is more about stimulating things like team science, and really working, supporting colleagues in their careers, and of course, we've been pushing from the start on and learning from working together with the other institute deans. But it has been a challenge in the pandemic, because indeed, nobody wants to do the first thing they do is actually close the doors and to say to colleagues in the institute, I'm sorry, but you cannot come in because we close your laboratory. And that has been very challenging. It's definitely held us back in the development of a positive, let's say, institute and atmosphere. I think, I'm always a positive person. So I think one of the good things is that the whole online working has been very, very beneficial. So if we have a NUBI PI meeting, we frequently have 80 to 90 people on a call. But you cannot imagine a face-to-face meeting where 90 colleagues would come up every month for a PI meeting. So I think the online working, I think we've learned a lot from, and I think we see a lot of benefits. So that is something which I see as positive, and people have come to realize, that you can do a lot from home, and actually also, of course, I'm also involved I have also a role in the faculty as Dean of Global. Of course, international collaboration, actually, it's just as easy to have a Zoom call with somebody in Australia as it is with somebody from Newcastle. The only thing is the time zone difference.
Yeah, sure.
But I think that's also something that I very much want to, I think people need to realize how easy it is to set up collaborations nowadays through technology. So I think there is a lot there. That's not only important for our research, it's also for our teaching. So we can invite world famous colleagues to give a seminar in one of our modules. So I think those things are quite nice. I mean, I've done some things in India, and it's very easy nowadays. You just you give your talk, and I think we haven't realized yet all the potential out of that.
Sure.
But at the same time, I'm extremely happy now to be back on campus and to meet people in person. We had a faculty board meeting this morning with everybody there. And it's just so important to see colleagues, and we need to go back to this period. I hope that we can somehow get it blended working in a good way. That will be a big challenge.
I think so. I definitely agree with you about the kind of, again, that barrier to collaboration being brought down significantly. It's so much easier to find to find a big group of people and find a time that works for everybody, and because they don't need to be somewhere that doesn't take 2 hours out of your day traveling around, to a different part of the country or a different part of the university or anything like that. So yeah, the kind of getting everybody together in one space is much easier virtually. But at the same time, you do miss something from being in person and meeting people at another meeting, and that kind of collaboration just after or just before a presentation, or trying to talk to people just after they finish giving their presentation to pick their brains a little bit about it is something that is harder to do. It's not impossible to do on Zoom, and certainly harder to do. Actually, one of the things that I wanted this podcast to serve as was just that, to kind of serve as that, to try and talk to people who are going to give a presentation to the IMA theme and other themes and other people in the university, and trying to provide that general background to them, and because that's the interesting bit to me, as well as the research talk, is the other things that they do that might be invaluable to your work. I think that's a really valuable thing to try and push.
I mean, I absolutely agree. I mean, I'm chair of a program committee for the European Society of Human Genetics. I've organized online conferences the last 2 years, and they've been very well attended. We had record numbers of colleagues attending online. But it is true that after a conference like that, you feel like, who did I really meet, that I got a real new collaborative contact? And I think that's something that we all know how important it is to meet colleagues after a talk, have a chat, perhaps have a drink, or have some further discussions. And I think especially for the early career researchers, that's so important that people can go back to meeting people in face to face. We will travel less. I think the climate change is important. We will, I mean, I've have to say that I've traveled much too much in my career, and I'll definitely travel less. But I think it is important still that colleagues sometimes, once or twice a year go abroad and see colleagues.
Yeah, for sure. I think there may be a push. I mean, I don't know how you feel about it. There may be a switch to more local conference attendance as well. So there may be more people willing to go to smaller conferences, or conferences that are nearby or in the same country, and then go to those bigger conferences online, and to get both of the experiences. But like you say, it's the experience of going to a massive whole international conference is not one you get very often, and it's really hard to replace that, and to change the contacts you meet there.
No, and I mean, I don't know about you, but I mean, I have to be honest. For me, that has always been one of the fun parts of my career. Is to travel and to meet colleagues. I have to be honest that is I mean, this Society of Human Genetics, the European Society, is a bit like my family, right? So it's if I meet them at the conference, it's really, I mean, that's something that you build up over years, and I think there's quite a lot of colleagues like me who have that. They build up this kind of links, and they're extremely important for your career, but especially just for yourself. Just one thing that you do. So yes, it is good to have these. I think at the same time, I mean, now that we're going back to face-to-face meetings, hopefully a bit more, what I don't want to do is lose, let's say, the online contacts as well. And if we have people from Latin America and Africa now joining this genetics conference online, very cheap and very affordable, next year they should also be able to contribute and join. And I think that's the challenge in the blended working that we need to tackle in the coming years. Yeah,
I think there's hopefully there'll be a best of both worlds scenario that can be settled on. I think that's definitely ideal. I just wanted to ask you, actually, because I don't have a lot of face-to-face time with kind of dean level people, so I'm gonna pick your brains on a couple of things. I know you've mentioned one of the things you were focused on when you took over the role was cross-theme communication. So getting not just a top-down approach from whatever the faculty decides. You don't want that to necessarily dictate what people do in their themes, but you want the themes to not just exist as silos as well. How do you think we can kind of encourage themes to talk to each other? How do you think, from our perspective, from my perspective, the IMA, because we're kind of cross different institutes, we have a lot of different people from all different walks of the university kind of in the same pot. But the other themes, maybe they only have people from biosciences in there. And how can we kind of generate more people talking to different people in different themes? Do you think there's a good model for that?
I mean, yeah, I think a lot is happening already. Yeah? So you see various teams seeing, let's say, overlapping interests and opportunities. So some of these things are just happening naturally. I think as an IMA team, of course, technology is something that is interesting to most colleagues in the different institutes, definitely in our Institute of Biosciences. So I mean, this is a question of just reaching out and saying, hey, shall we do something together? And we keep on promoting that when we have discussions with the theme leads regularly. We look at that. What we don't want is that the themes become the new silos, the new institutes, basically. Yeah. So we definitely want them to be open. But on the other hand, some themes are very big and are very occupied with just getting that organized in a good way. So there is not one way to do it. Some themes are quite small, so we encourage them, go out and talk to other themes a bit more, and learn from each other again. In the end, it's individuals talking to each other, and yeah, I think it's working relatively well, but we can always improve. But yeah, I do see more and more people talking about joint team meetings and seminars and stuff. So.
Sure, I think so. Yeah, I'd like to hope we can get kind of more people from different aspects. Kind of, certainly when it comes to the technologies that people use, because a lot of the time, those kind of common grounds are the technologies that people use. People use them in different ways from different research projects. So something that we're trying to kind of push forward at the moment is the special interest groups, which can be open to anybody from any theme, but they're focused mainly around an application or a methodology, and what different people do with that different methodology in their research groups that might be of interest to other people using those. So I really hope those are useful, and they certainly seem to be useful. But the ones that we have set up now are certainly useful to the people in them. But yeah, I really hope those can be something that we can drive a bit more collaboration through as well. That'd be really good. So lastly, one of the last things I wanted to talk about is because one of the other interests of mine personally, and I know the core facilities in general, is public engagement and outreach that we have going on. And it's certainly something that I personally want to do more of. And I know it's something that the Bioscience Institutes takes great pride in, because the website's got some really good examples of successful outreach that we've done. Do you have what kind of plans have we got coming up for public engagement and outreach within Biosciences in the near to mid future? Is there anything in the pipeline that's kind of really exciting, or is it anything we can engage with school kids or students or members of the public that you want to promote, or are you open to new suggestions?
I'm very open to suggestions. I think most of the outreach is this is not a top-down thing that we as a Bioscience Institute say, now we're going to do public engagement. And so often what happens is that this is done, people have certain funding, they work together with certain charities. So this is how things work in part. Then you have colleagues are being asked to give, for example, a talk at I did a talk a few years ago on the Pint of Science. So these kind of engagement things. So there is not a systematic thing of we as the institute trying to stimulate public engagement, but I think it's something that's very useful to do, and it's something that actually it makes you also, if you want to think about, well, for example, if you want to write grants and be successful in that, you need to be able to explain to colleagues why it's so important. So one of the things, even if you think I'm very busy and I don't want to do public engagement, can actually help you write better grants. And for example, at the Center for Life, they even have people who can help you with doing performances and to speak in public speaking and all these kind of things. So I definitely would promote colleagues to do that. Giving talks to lay audiences has helped me a lot in better, let's say, explaining to colleagues. And also for myself, better understanding why actually do I want to do this?
Yeah, absolutely.
We don't have a systematic, let's say, discussion on this in the institute. I think it's a good idea. We could pick this up in the management team. So we're always interested in ideas. Perhaps we should do a day of outreach, let's say the top, let's say, engagement activities from our institute. We don't have that planned yet, but could be nice. Could be, for example, after NUBI live as a next thing to do. Yeah.
That sounds like a really good idea to me. I think you're right in saying it can add a lot to. It can be really beneficial to your research. It's very easy to kind of get lost in the technical and the small of your project and not lose sight of the big and the overarching why you're doing what you're doing, especially if you're like a PhD student. I certainly felt like that when I was doing my postgraduate studies, that it's so easy to get hyper focused on trying to solve that one tiny problem that you've got, that actually taking a step back and working with people who are just interested in it, or just want to learn a little bit more and maybe just want to, maybe it's people with a rare disease. I've certainly done some volunteering for blood cancer charities, and the feedback you get, the feelings you get back from it are nothing but beneficial. It feels really, really nice to talk to these people.
Yeah. No, as you say, this is not only that it's good for your career. It's actually just, it is something that I, very much enjoy. I mean, when I speak to patient organizations, it's something that, you feel like, they are really interested in what we do. And then, I think it's very important. Yeah, it's part of what makes me want to be a scientist.
Yeah, absolutely. And also you're right. It does help with framing your questions or future questions that you've got in such a way as to appeal to people reading your grant applications, because not everybody reading your grant applications maybe knows, well, theoretically they should know everything about what you're applying for, but it's new research, right? You need to be able to frame it in such a way to make it applicable, to make it clinically relevant, to make it, to put all these things in. And correct me if I'm wrong, I haven't applied for funding for a very, very long time. Are more funding bodies interested if you do put like outreach or public engagement aspects of funding?
That's very important. Yeah,
because I'm aware that some funding bodies have a percentage of the funding given to you to go towards kind of public engagement and outreach, but it's definitely shouldn't just be a tick box thing. It should be.
So like, I mean, I was in a panel for the Wellcome Trust last week, and I had to basically look at about 20 grants, and they all have a part on public engagement. But at the same time, I mean, you say that you understand everything, but when I read 20 grants on genetics, actually I can only understand part of
Sure.
these grants. And I think one of the things that I would urge everybody to do, and you learn that by public engagement, is actually telling a story in a simple way. And I think 90% of colleagues still tell the story too complex.
Yeah.
And then people like me, I'm not the most stupid, but I'm also not the most smart. I need to make a decision on your grant. So try to put grants at the level that an average person who's smart, can understand it. Yeah, I think that's something that most colleagues have quite some challenges with, and then they struggle. They say, why didn't I get the grant? And partly it's because you've written it in a quite complex way, and we as panelists have to then go through all these grants and try to judge them quite quickly, right? I mean,
that's the thing to bear in mind, isn't it? That you've got a finite amount of time and a finite number of grants. So if you're getting a small fraction of your days or your weekend or your weeks.
It's very rare. It's very rare that I have almost never seen a grant where I think this is put too simple. It's too easy.
Oh, that's very interesting.
90% of the time it's too complex.
Yeah.
So I would say make it more simple, and that doesn't mean dumb it down, but explain your ambition, explain your theory for an educated audience.
Yeah. I think that's a really good thing to bear in mind when you're writing, and not just that. It doesn't hold true just to grant applications. That's also true of if you can introduce a paper that way, it makes it all the more engaging to read. If you can write an abstract that way, so many abstracts that I read, I'm sure it's the same for you. It's less of a brief summary of the work and more of how many abbreviations and technicality can I get in a short paragraph. And it should be, I can read the results if I want to find out everything about it, but I want to read why you did it and what you found in the abstract. So that's a really good point. Definitely one to bear in mind. So just as we're wrapping up at the end, I generally try to ask a similar sort of question to most people I interview. So what I want to ask you is, what can IMA members specifically, or people listening to this podcast, what can we do to contribute more to what you have in mind for NUBI? What can we tell people? What do you want to tell people to do if you have an audience behind us listening to this? What can we drive them towards? What can they do?
Yeah, I mean, I think, clearly I'm a technologist, right? So I think we should be very proud of our technical expertise. And I think it is about, thinking about how can we work together to use that expertise in the best way. So I think everybody's doing their utmost. So I'm not going to say you need to do this or that, because I think people are doing the best they can. But it is, one of the things that I find very important is that we give more freedom to technologists to apply for grants, to come up with ideas. And I think that was something that's been, academia has been very conservative, but I think university here in Newcastle is quite open in allowing colleagues to go for grants and take initiatives. And no matter whether you're an RA or a technician, or whether you're a professor, just come up with ideas. So I think, don't feel limited, and just go for things and discuss things with colleagues, come up with ideas, and yeah, let's just do this. So that's what I would do. Just don't feel limited because of your role or position. But if you have ideas where you think, this is really important, discuss it. I think that's one of the nice things. Go with it first within your theme. But sure. Send me an email. I mean, it's very rare that I get an email with somebody saying, I have a good idea. What do you think of it? Whereas, and these are the most fun emails to get, right? Yeah. So I get lots of emails, but I rarely get an email with say, I have this idea. What do you think? And I would love those emails. And but so yeah.
I'll bet that it might. I'll make sure everybody tells me a good idea. I'm going to give them your email address, and then hopefully we can flood your mailbox with good commercializable ideas. Okay, well that's excellent. I've taken up more than enough of your time. Thank you so much for talking to me. I'll wrap up at the end here. Just saying to people listening that we've got the IMA team's got some really wonderful talks coming up. We've got a presentation from Claire Stevenson, who's a scientist at the John Innes Centre, who does some fantastic work championing technicians and their role in research, and she's going to be talking to us a little bit about that. And then an Intel talk from Pauline Addis, who is the IMA's contact for the FMS Faculty of Medical Science Impact Officer. Sorry, yes. One of the feedbacks I got from these podcasts, too many acronyms. So I'm going to start explaining what my acronyms are. It's so easy to fall into those acronyms, the acronym speak, right? So yeah, a couple of talks then. I've got a podcast episode coming up with Claire Stevenson, so that'll be published just before her presentation, and I've got one with Pauline and Callum, her colleague, as well. So I'm going to try and pair these up just prior to people coming to talk to the theme, and hopefully we can get some more interest get people coming along and to chat to them and ask them questions based upon what we hear here. But yeah, thanks again for talking to me, talking to you, Joris. It's been really nice to talk to you, and good luck with everything. And if I do have any good ideas, I'll be sure to email them through.
Thanks very much, Andy. It was a pleasure talking to you.
Lovely. Thanks very much. Bye now.
Bye-bye.